Hi, good afternoon, everyone. Thank you for joining us for today's webinar. I'm Cassie, Marketing Supervisor at GenScript, and I'll be the moderator for the webinar today. A little bit background about GenScript for those who are new to us. GenScript is the leading life sciences research and application service and product provider in various fields, from basic life science research to translational biomedical development, industrial synthetic products, and cell therapeutic solutions. The topic for today's webinar is Cell and Gene Therapy Solutions in Cancer Therapy Research. And we are happy to have our field application scientists, Dr. Edward Wong and Dr. Leung How Wing, as the speaker for today. So let me introduce you our first speaker for today, Dr. Edward Wong. He's the field application scientist covering GenScript molecular biology portfolio. He completed his PhD degree from University of Adelaide and worked as a postdoctoral fellow at National Cancer Centre, Singapore for 3 years. So during his time in NCCS, he reported the predictive factors for genetic screening of BRCA1 and BRCA2, and also the importance of genetic testing for 25 breast cancer predisposition genes using NGS technology. Our next speaker for today, Dr. Leung How Wing. She is the field application scientist supporting the protein expression and antibody production services at GenScript. Dr. Leung graduated at the University of Singapore in 2011. Majoring in pharmacology and neuroscience. She has more than 10 years of research experience in molecular biology techniques and extensive knowledge in protein structure modeling, protein expression, and its purification. This webinar will take around 45 minutes, and we'll have about 15 minutes later for Q&A session. You can type in your questions in the chat box, and we'll answer them after the presentation. If we didn't get to answer your questions during the Q&A session, we'll be sure to email the answers from the speakers back to you after this webinar. So without further ado, we'll let Dr. Edward to start his presentation now. So Dr. Edward, over to you.
Good day, everyone. It's a great pleasure to be invited in this webinar. And thank you so much, Casey, for the introduction. So as my colleagues introduced, I am Edward, the field application scientist from GenScript. And today, I will be taking this opportunity to share about the potential use of CRISPR for therapeutic purpose. So these are the contents that I will be presenting, including a brief overview about our company, the CRISPR introductions and its medical applications, followed by the collaboration case study between GenScript and our client to share with you an idea about potential use of the CRISPR in immunotherapy. And lastly, I will wrap up my presentations by demonstrating what GenScript is capable of in CRISPR. Now, to begin with, I would like to briefly introduce the history and milestone that our company has achieved throughout the years. So our company established in the year of 2002 in New Jersey, United States, and was the first company to commercialize the gene synthesis. And GenScript has achieved many successes throughout the years, particularly in the year of 2020. Since the outbreak for SARS-CoV-2 virus, GenScript has successfully been co-developing the CPAS neutralization antibodies detection kit with Professor Wong from Duke NUS in Singapore. Moreover, our subsidiary company, The Legend, which collaborates with Janssen to co-develop and co-promote the CAR T-cell therapy for myeloma disease, was also listed on the Nasdaq and China stock market. Now let me walk you through the history of gene therapy development to understand why it is important to personal and precision medicine. The introduction of gene therapy into the clinical use provided the hope for thousands of patients with genetic diseases. Initially, the gene therapy utilized the viral vector delivery for the therapeutic transgene for the cancer treatment or the monogenic disease. Now, the first clinical trial that used the gene therapy for therapeutic purpose was approved in the year of 1990 for the monogenic disease known as the adenosine deaminase severe combined immunodeficiency, or known as the ADS-SCID. In two young girls with the ADS-SCID disease, were treated with the retroviruses through the ex vivo delivery of a wild-type adenosine deaminase gene to the autologous T lymphocytes, which were then infused back into the patient. While one patient showed moderate improvement, the other did not. But the future of gene therapy was hampered by a major failure in the clinical setting. In the year of 1999, an 18-year-old boy, Jesse, who received the genetic treatment for the OTC deficiency disease, died from the treatment as a result of stronger immune response triggered by the adenovirus vector, causing a chain of multiple organ failures. And shortly after, another gene therapy trial on skip disease also shared the same fate. So after this setback, the researchers have been working on improving the safety of gene therapy by re-engineering the virus before the clinical trial. Alternatively, the non-viral vectors have been used as an alternative method for gene delivery, which have reduced immunogenicity. The discovery and development of the CRISPR system has provided a second opportunity for gene therapy to recover from its stigma. In 2020, the Nobel Prize in Chemistry has been awarded to Dr. Shapunti and Dr. Ssdna for discovering the CRISPR system. So the discovery of this tool enabled scientists to simplify genome engineering. Additionally, this approach provides a possibility to correct the existing genetic aberrations. This alternative approach would allow the pathological mutations to be repaired while avoiding the risk of insertional oncogenesis in order to fulfill the concept of precision medicine. So the revolutionization of this technology has inspired the researchers to solve the problem using CRISPR. The CRISPR system harnesses a few components from the adaptive immune response that's found in the bacteria for genome editing purpose, including a guide RNA and a Cas protein. Please note that the guide RNA that I show here in this picture indicate a single format of the guide RNA that consists of a 20 nucleotide of the cell RNA sequence that recognize the targeted region and a 67 nucleotide of the tracer RNA that helps to track the Cas endonuclease to the targeted sites. And after the formations of this CRISPR-Cas complex, it can then trigger the cutting ability to form the double-strand break. There are many types of the Cas endonucleases out in the market, and the Cas9 is the most commonly used to date. So the double-strand break can be repaired through two different pathways: the non-homologous end joint pathway, or known as the NHEJ, and also the homology-directed repair pathway, or known as the HDR. In brief, if there is no donor template, the repair will occur by the error-prone NHEJ pathway. And as you can see here, this repair approach basically will delete the random nucleotide around the breaks, therefore resulting in the indel that cause the frame shift and subsequently knock out the protein functions. On the other hand, when there is a donor DNA template that flanked by the long homology arms on each side, which is the 5' and the 3' end cell, that is complementary to the break, what the cell will do in the first place is to incorporate this fragment into the genome because the homology arm is already there. And with this method, it allows us to introduce point mutations, gene knocking to the site, or introduce a tagging and even a gene correction. So with the understanding of this system, studies have been conducted to determine the usefulness of this system for gene therapy usage. Here I listed a couple of examples. In the cancer therapeutics, this technology has been used with several types of the cancer research, including the breast cancer, the endometrial cancer, lung adenocarcinoma, and prostate cancer. And coupling with the research, the mouse models for each of the cancer disease have also been generated. Additionally, in the frame of genetic diseases, the establishment of this technology provides a useful tool to make the idea of gene correction become feasible. And these diseases include the hemophilia A, the cystic fibrosis, Huntington disease, as well as the cardiovascular disease. Additionally, as the interest in immunotherapy grows, the researchers is in the search of a better tool to engineer the T cells for clinical usage. Currently, as we all know that the T cell engineering requires the use of the viral vectors as a result of its high efficiency. But its high off-target rate that cause the cytotoxicity and the genotoxicity are also the two main concerns to the researchers as well. Therefore, the researchers are in search of a better tool to overcome their limitations, and CRISPR seems like an ideal way to engineer. I will further elaborate in the following case study. In the following slide, I will be presenting a case study to demonstrate the research outcome started by GenScript and our collaborator on the use of CRISPR for T-cell engineering. Now, this collaboration was between GenScript and Milestone Laboratory from the University of California in San Francisco. In this article, the author described the use of the recombinant viral vector to genetically reprogram the T cells for therapeutic purpose. They may be hindered as a result of its high off-target rate. Additionally, the need of the viral vectors has also slowed down the research and the clinical use because their manufacturing and testing is lengthy and expensive. But the discovery of genome editing brought the promise of specific and efficient insertions of large transgene into the target cells using the HDR repair pathway. And the researchers proposed a non-viral approach using the CRISPR to insert a large DNA sequence at the site of interest rapidly and accurately, while preserving the cell viability and function. And with this technology, the author further studied the feasibility of using the CRISPR to modify the individual or the multiplex endogenous genes. So in this article, the author demonstrated to us how he studied the potential use of CRISPR in reprogramming the T cells through a systematic experimental design, and also provide a concrete data analysis to obtain the proof of concept which CRISPR is the best toolkit to overcome the limitations with the use of a viral-based genome editing approach. These objectives include the investigations of the efficiency of the non-viral genome targeting in the primary T cells, the applicability of this system in different locations throughout the genome, the on-target and the off-target rates of the double-strand DNA integrated sequence in the targeted locus, and the efficiency of a single-strand DNA as a HDR donor template in place of the double-strand DNA in genome editing. And lastly, the potential use of CRISPR for therapeutically purpose. So a major barrier to effective non-viral T-cell genome targeting of large DNA sequences has been the toxicity of the DNA. Although the introduction of a short single-stranded Ssdna as a homology-directed repair donor template does not cause the notable T-cell toxicity. It has been shown that a larger linear double-stranded DNA template are toxic at high concentrations. So as displayed in this data, the black line and the gray line indicated a high cell viability where Ssdna was used as a donor template for editing, whereby the use of either plasmid DNA as indicated in the red line, and also the double-strand DNA as indicated in dark blue color, demonstrated that these materials possess the toxicity to the cell. But the author found that the core electroporations of the human primary T cells with the CRISPR-Cas9 ribonuclear proteins, or known as the RNP complex, with the long linear double-strand DNA template, reduce the toxicity associated with the double-strand DNA template. So based on this finding in this study, the Cas9 RNP system were co-electroporated with the double-strand DNA HDR template to introduce the N-terminal GFP fusions in the housekeeping genes known as RAB11A gene. So this diagram here illustrates the genome targeting workflow. The genome editing reagents, including the guide RNA and the HDR template, were carefully designed and synthesized by GenScript. And 2 days before the electrophorations, the primary human T cells isolated from blood or other sources were stimulated. And on the day of electrophorations, the guide RNA complexed with the Cas9 endonuclease protein to form the RNP, and also the HDR template, as well as the collected stimulated T cells were mixed and electrophorated, in which a process taking about approximately 1.5 hours. And after the electrophorations, engineered T cells can be readily expanded for an additional 1 to 2 weeks. The viability was measured 2 days after the electrophorations, and the GMP expression was measured at day 4. So the picture here shows the percentage of the GMP insertions into the endogenous RAB11A gene in the primary CD4 as well as the CD8 T cells. As you can see that with the use of the RNP together with the double-strand DNA donor template, the result demonstrated a GMP expressions in a near 50% in both primary and both human primary and CD4 and CD8 T cells. So additionally, the results show an average efficiency with the RAB11A used with the GMP template at about 33.7 and also 40.3% in the CD4 and CD8 cells respectively. Compared to the non-electroporated control, the cell viability study indicated that the survival rate at about 68% after the non-viral genome editing workflow. The author next confirmed that the system could also be applied broadly by targeting the sequences in different locations throughout the genome. To do this, they efficiently engineered the primary T cells by fusing the GFP with the different locations, including the CD4, CLTA, ACTB, FBL, and tuber 1B, and demonstrating its specificity. A live cell imaging with confocal microscopy confirmed the specificity of gene targeting, revealing the distinct subcellular locations of each of the resulting GFP fusion proteins in the endosomes, the plasma membrane, clathrin, actin, nuclei, as well as the microtubules. Moreover, the author had demonstrated an achievement in fusing the RAB11A gene with either the GAP as well as the M-carrier protein, followed by the success in creating the biallelic tagging with both GAP and M-carrier at the same locus, or even multiplexing in 2 or even 3 different locations, indicating several endogenous genes can be directly engineered without the virus in T cells. So for therapeutics use of the genetically modified T cells, integrated sequence should be introduced specifically without unintended disruptions of the other critical genome sites. So in order to understand the target specificity, the author conducted a study in which to insert the GFP proteins at the N-terminal of the RAB11A gene. So the homology arm specified the exact sequence where the insert, which is a GFP tag, will be inserted, following for the scarlet integrations of the exogenous sequences. Now, because a GFP fusion protein is created, the GFP fluorescence will be seen as a result of the on-target integrations, which is dependent on the RNP cutting at the integration site. On the other hand, to investigate the unintended non-homologous integrations using the RNP system, or construct that consists of the N-terminal GFP RAB11 fusions and endogenous of the RAB11 promoter within its 5' homology arm was constructed. So this construct would express the GFP at the off-target integration site, which allow us to assay for the off-target events using the flow cytometry. Additionally, a guide RNA was designed to cut a genome region that is not the homologous regions to the targeting sequence that can be used to infer integrations at an off-target cut site. So you can see here, by quantifying the GFP positive cells, an increase in the green fluorescence level at near 39% was observed, indicating the success in the on-target integrations. But surprisingly, there was a near 1% of the off-target rate observed among the cell pool. A similar experimental design was conducted across different regions, including the CLTA, Tuber1B, ACTB, as well as the FBL as shown in this picture. The black colors indicate that the template alone was electroporated, and the red color indicated an off-target RNA along with the template was an off-target RNA that was not the homologous sequence along with the HDR template was electroporated, and the green color indicated an on-target guide RNA along with the HDR template was electroporated. So the increase in the percentage of the fluorescent cells over the limit of detections when the template alone was electroporated probably represented a random integration at the naturally occurring double-strand breaks. But not every off-target integrations will yield the fluorescent protein expression, such as the case of the Tuber1B here. Now, this could be a result from only part of the template sequence could be integrated, or it could be integrated in a way that does not lead to a measurable expression. And as expected, the efficient GFP expressions was only seen with the correct guide RNA sequence, and the HDR template was co-electroporated. But the author had also found evidence to suggest the double-stranded templates could integrate independent of the target homology, although at a low rate, as shown in the red color here. Nevertheless, for the sake of the safety in therapeutic usage, this data suggested that it is imperative to seek for a different format of the HDR template in order to mitigate the off-target issue. So in order to overcome this limitation, several studies have been conducted to assess the feasibility of using the long Ssdna template to reduce the homology-independent integration while preserving the on-target efficiency. But in the past, when a chemically synthesized technology was immature, the productions of a single-strand DNA had been complicated. The two commonly used approaches for the productions of the Ssdna include the use of the exonuclease to degrade one strand of the PCR product that had been labeled with the 5' phosphorylation, or through the IVT-RT approach, in which generations of a ssRNA template followed by the synthesizing of the cDNA through the reverse transcriptions and subsequently degrade the RNA. But now, with the advance in technology, GenScript possesses the capability to chemically synthesize a long KB of Ssdna efficiently. The result here demonstrated that Ssdna achieved a similar, if not better, knock-in efficiency compared to the double-strand DNA, and the efficiency increased with the increased amount of the templates was being used. Additionally, the further analysis demonstrated a higher cell viability with the use of the Ssdna as shown in the blue bar here compared to the green bar, which was indicate the double-strand DNA was used. And importantly, the off-target rate was significantly reduced with the use of the Ssdna compared to the double-strand DNA template. So while all of this data presented above demonstrated the potential use of a CRISPR technology to rectify the mutations and the integrations of a much larger DNA sequences to the targeted region specifically, it is still not known of its efficiency in reprogramming the antigen specificity of human T cells, which is critical for many cellular immunotherapy applications. So a number of work demonstrate that the chimeric antigen receptor, which is known as CAR receptor, has enhanced efficacy when they are genetically encoded in the endogenous TCR locus using the Crispr system that delivered using an AAV-based approach. But to target a specific TCR sequence to this locus is a more challenging problem because the T cells must express paired TCR alpha and a beta chain to make a functional receptor. So in this article, the author developed a strategy to replace the endogenous TCR using the RNP system, as described earlier, to integrate an approximately 1.5 kb of the DNA cassette into the first exon of the TCR alpha constant regions, or known as the TRAC. So this cassette encoded the full length of the TCR beta separated by a self-exciting 2A peptide from the variable regions of a new TCR alpha, which can encode the full TCR alpha sequence when appropriately integrated into the endogenous TRAC exon. So to test this strategy, the author introduced the TCR beta and the TCR alpha that recognize the NY-ESO1 tumor antigens, or here let's call it an NE1 beta in this context. So into the TRAC locus of a polyclonal T cells isolated from the healthy human donors. The antibody standing for the total TCR alpha and beta expression, and also the NE1 beta MHC dextramer standing for the NE1 TCR expression, revealed that a 12.3% of the engineered cell carried the correct construct. Furthermore, the non-viral genome targeting enabled the reproducible replacement of the endogenous TCR in both CD8 and CD4 plus primary human T cells. The T cells that did not express the NE1 TCR were probably the TCR knockout. Presumably, due to the non-homologous end joining events induced by the Cas9 mediated double-strand DNA break in the TRAC exon 1. So next, the author assessed the tumor antigen-specific functions of the targeted human T cells. So when targeted T cells were co-cultured with two different NE1 antigen-positive melanoma cell lines, known as the M257 and M407, the modified T cells robustly and specifically produced the interferons gamma, the TNF, and also induced the T cell degranulations, or known as CD107A, shown right here in this picture. So the cytokine productions and the degranulations only occurred when the NE1 TCR T cells were exposed to the cell line expressing the appropriate NE1 peptide on the MHC to the TCR receptor. So the outcome was comparable to the response T cells from the same healthy donor in which the NE1 TCR was transduced by the gamma retrovirus and expressed using a viral promoter as shown in the black bars here. So the killing assay was also conducted to measure the cell death of NE1 presenting cancer cells to the TCR expressed T cells. So that, as you can see from this picture here, the cancer cells reduce significantly when co-cultured with the T cell that express the correct TCR receptor. So this study provides evidence that the discovery of a CRISPR provides an alternative tool for the engineering the T cells for therapeutic purpose. Furthermore, the RNP system helped to simplify and accelerate the entire genome editing workflow, with the approach being optimized using the Ssdna as a DNA donor template. And looking forward, this technology could be potentially used to rewire complex molecular circuits in the human T cells, and multiplex integrations of the large functional sequence at the endogenous loci should allow the combinations of the coding and the non-coding elements to be corrected, inserted, modified, and even rearranged. So for the audience who has a strong interest in understanding about this work, Dr. Rolf has previously gave a presentation sponsored by GenScript. You may either access to GenScript website and look for his webinar, or you may also access the webinar through the link as shown right here. So before I wrap up my presentation today, I would also like to take this opportunity to introduce the GenCrispr services and products that we are offering. Now, with the experience and a team of experts in CRISPR, GenScript extends its arm and offers a wide range of GenCrispr services to assist the research communities in their project. These services and products include the plasmid or virus-based or CRISPR construct, the components required for RNA system, including the sgRNA, double-strand DNA, single-strand DNA, or Cas9 endonuclease proteins, the CRISPR libraries, CRISPR cell lines, and even a CRISPR microbial editing service. So I thank you for your attention.
All right, now let's welcome Dr. Leung to give us the insights on how peptides can aid in the delivery of genes to targeted cancer tissues and also different ways you can identify neoantigens that are specific somatic mutation expressed only in tumor cells. Dr. Leung, over to you.
Hi, everybody. Thanks, Casey. So for the nice webinar. Good afternoon. Thanks for taking time off your busy schedule to attend the webinar, and also for staying for the second part of the talk. I'm How Wing, and I'm the field application scientist for GenScript, and it is my pleasure here to share the current trend in the industry in ways to enhance cancer treatment by using peptides and neoantigens in gene and cell therapy. Just a moment. Okay. Okay. Okay, just a moment, because I have some problem trying to move to the next slide. Okay. Okay, yeah, maybe I'll use without the laser pointer then. So for today's talk, I will first share some cancer statistics and also the pressing need for effective and better cancer treatment. What is gene and cell therapy and how peptides and neoantigens can be effective in promoting gene and cell therapy. In addition, case studies will be shared for illustration, and also I'll be briefly sharing some of the capabilities of GenScript in neoantigen peptide synthesis. So as of 2017, cancer is the second leading cause of death in the world, affecting nearly 9.6 million. And although many years of research have been poured in with lots of time and money invested and spent on cancer, we still haven't cured cancer. And why is that? So to put things into perspective, not all is in vain. There is a better survival rate from having 25% of people with cancer that will survive 10 years or more after their diagnosis in 1970 to 50% now. So the difficult issue of getting a cure for cancer is that cancer is not just one disease. Instead, it is an umbrella term for more than 200 distinct diseases with distinct ones affecting even the gender itself. So moreover, each broad cancer type has many subtypes, and they all look and behave differently because they are of a different genetic and molecular background. This is because as cancer arises from our own cells, so each cancer can also be as different and diverse as people are. So, however, currently the most common type of cancer treatment has still been surgery, that is to remove tumor. If the particular cancer type is sensitive to radiation therapy or chemotherapy, one would receive one of these therapies as the primary treatment. And due to a myriad of cancers, other therapies such as stem cell transplants, and hormone therapy are available. However, as the genetic mutations that cancer arise acquired over the time means that the cells change the way they behave. So this can be incredibly difficult during treatment, because the mutation can lead to cancer cells developing resistance to a treatment over time, making it ineffective. So in recent years, gene and cell therapy has been gaining traction as they present a more targeted gene alteration and also a training of the patient's own immune system to fight cancer. So what is gene and cell therapy in cancer? Well, gene therapy involves the replacement of a defective gene with a functional and healthy copy of that gene, and is a potentially beneficial cancer treatment approach, particularly over chemotherapy. That often lacks selectivity and may cause non-specific toxicity. Cell therapy is an adoptive cell therapy, tapping on the cells in our immune system to recognize cancer cells as foreign and eliminate them in the process. However, though the promise of accuracy in pinpointing and correcting the mutated gene in cancer, there are limitations in gene therapy, and one important roadblock is their delivery. Due to the anionic properties of the gene itself, there are poor translocators into the cell membrane. In fact, only 10% of the genome can be targeted by the conventional rationale of the Lipinski rule of 5 for small molecule design. So this make the delivery of nucleic acid to the correct site of action very inefficient. And recently, we have, as of date, there are several types of delivery. For example, through the virus, and also non-virus factors such as ionic polymer, dendrimers, CP peptides, and also liposomes. And although the most common gene therapy factors are viruses, they are still concerned with the toxicology profile, such as adenoviral stimulation of the immune responses. In addition, such genes has to be made cell specific and has to have an ability to penetrate intracellularly and are not vulnerable to enzymatic degradation. So as mentioned in my earlier slides, so peptides demonstrate one way of non-viral delivery of genes, also called the cell-penetrating peptide CPP. They are majorly divided into non-cell specific CPP and also cell-specific CPP. For non-cell specific, there are three main types, the cationic types, such as the TET derived from the HIV or the NTP. These are rich in arginine and lysine and make them cationic. Not only for DNA, they are effective as a transduction peptide. For example, a polymer of more than 12 amino acids have a better effect in transduction of the virus. So there are also the amphipathic non-cell specific CPP, in which they are chimeric peptides attaching the hydrophobic domain to the nuclear localizing signal through a covalent bond. At times, amphipathic peptides could simply be a signal peptide too. So for cell-specific CPP, usually these peptides are identified through screening of peptide libraries, and because of the higher specificity, they are needed in smaller doses compared to the non-specific CPP, which are usually not so good for brain or poorly vascularized tissues. An example of such screening is for this peptide TIT7, which is a linear 7-mer peptide screened using the phage library that could bind to the endothelial surface similarly to a carbohydrate moiety. So from the earlier results, they noted that Nonaka and all noted that the stability of this peptide to protease, the instability of this peptide to proteases, and aimed to use D-amino acid to increase its stability. So instead of just replacing all the L-amino acid to a D-amino acid, which sometimes are not accurate and ineffective, they use a mirror phage library and identified the best targets that do not bind to normal tissue. And using the peptides generated in GenScript, they set up the library assay, find the optimized stabilized peptide, and validated its transport towards the targeted brain tissue. And transport of the drugs guided by the peptide towards the brain also decreased in the tumor size, opening up the possibility of this peptide for other gene delivery. So in this next example, we also have a strategy that was used by Don and al. Published in the Journal of American Chemical Society to use the RGDK peptide for dual targeting of the tumor endothelium contributed by the RGD that interacts with the alpha-5 beta-3 integrin, which is overexpressed in tumor vasculature, and also by the RGDK peptide, which binds to the neuropilin-1 NRP1 receptor, which is present on tumor cells, hence promoting cancer cell penetration and uptake. And connecting the positively charged siRNA AD complex via the negatively charged polyglutamic E16, the peptide target the siRNA AD complex much better towards the tumor and is also able to slow the tumor growth much more effectively. And of course, besides those mentioned, there are peptides, other targeting peptides towards the heart, the bone muscles, for example, and also endosomal escape peptides to ensure gene carried are not degraded. Nuclear localizing signal peptides to target them into the cells, for example. So not only peptides are important as an aid for gene delivery, it also plays a large part in ensuring effective cell therapy. So what is cell therapy? So currently there are several kinds of cell therapy. Tumor infiltrating lymphocyte TIL therapy, where the T cells are already infiltrating in the patient's tumor, and they are harvested and activated to be expanded upon and reinfused back into the patients. The engineered T cell receptor therapy, the TCR therapy, where T cells are engineered and equipped with new T cell receptor to target specific cancer antigens that bind to the MHC. Chimeric antigen receptor CAR T cell rapid therapy, where the antigens need not be bounded by MHC and these antigens are expressed on the surface of tumor. And also natural killer cell therapy, where the NK cells are engineered with cancer targeting CAR to initiate the removal of antigen-specific tumors. And despite the great potential of using cells from the immune system to eradicate tumors specifically based on the antigen, there are some limitations of cell therapy, which includes, for example, how to ensure the proper activation of T cells, or to how are we able to maintain the T cell activity for enough amount of time to sustain effective anti-tumor response. And also how one could be sure that enough number of T cells are activated. Also, one has to consider the heterogeneity of the tumor itself and the possibility of a strongly immunosuppressive tumor environment. But most importantly, the specificity of the antigens towards the cancer could greatly affect the effectiveness of the cell therapy. In fact, non-specificity is also one of the major causes of side effects in cell therapy. So what antigens are we talking about? So there are different types of cancer antigen, which are potential as antigens for cell therapy. Tumor-associated antigens, TAA, are processed fragments of proteins, normally in low expression in other cells, but high expression in cancer cells. And these are usually used in targets in CAR T therapy. Cancer testis CT antigens are usually reproductive tissues antigens in reproductive tissues, but not in adult tissues, but appear in significant subset of the malignant tumors. Viral antigens are associated with a viral infection, and cancer cells express the proteins encoded by the viral open reading frames. And also neoantigen, which are mutations in non-germinized cell, and these somatic mutations accumulate in cancer for initiation, development, and also metastasis of the tumor, and are highly specific to tumors only. And of course, such antigens, especially the neoantigens, as non-synonymous mutations, they can be identified by whole exome genome sequencing, WES, from tumor and match normal DNA. The resulting epitope candidates can then be further selected based on their likelihood to be processed and presented on the surface HLA molecules using in silico prediction algorithm, or through a selection of mutated epitopes bound to the tumor cell surface HLA molecules through immunopeptidomics. And finally, a variety of novel high-throughput immunological screening methods with enhanced capacity to interrogate large number of candidates. Neoepitopes are used to screen cancer-derived CD8 positive and CD4 positive T cell population of interest for neoantigen recognition. To properly screen through the numerous potential mutations to identify the right neoantigen candidates, the method is crucial. As mentioned in my previous slides, there are several methods through in silico prediction, LC-MS, MS-based immunopeptidomics. All have the advantages and disadvantages. And such as for in silico prediction, we can greatly narrow down the number of candidates using the screening processes. However, many are algorithm or prediction based, and it is greatly dependent on the design and the accuracy of the prediction algorithm. They are also not optimal when dealing with MHC2 bound antigens, and considering the numerous polymorphisms in MHC2, this is more difficult for MHC2 bound antigens. And while LC-MS is high throughput and can detect mutant peptides and even non-canonical peptides, they only identify abundant peptides and are greatly dependent on the HLA expression on the tumor cells. So these are also there are also limited sensitivity sometimes from the mass spectrometry. However, if it is based on WES alone, although it can identify all candidate neoantigens, there is much screening involved, and it will have a limited visibility, especially for tumors with high mutation burden. So as such, sometimes multiple strategies are employed to better derive the neoantigen, as shown by Qian published here in Molecular Cell Proteomics, using MS-based proteomics and next-generation sequencing. They employed pan-HLA class 1 immunoprecipitation, illusion of the class 1 presented peptides, and high-resolution MS-based sequencing, and also NGS of the genomic DNA or RNA to create a patient and cell line specific databases incorporating variant peptides, and also computational algorithm to identify 35,000 HLA class 1 presented peptides containing 8 to 14 amino acid residues. In fact, using this strategy, they even identified 2 linked RNA derived peptides and their synthetic counterparts. They validated the binding here using the T2 cell-based stability assay, as they are HLA-02 binders. GenScript has also been involved in the generation of these peptides for the screening processes. So what about the tumor-associated antigen, TAA, that are often used in CAR T development? Here, Nan et al. Published in Cell Reports Medicine show an interesting strategy, targeting the glypican-2, an overexpressed protein in the neuroblastoma. They use RNA sequencing analysis and identify the exon 3 of the glypican-2 as overexpressed in cancer, but minimally expressed in normal tissues, obtaining a monoclonal antibody clone that binds specifically to the exon 3 and 10, shown by electron microscopy and also by epitope mapping. They managed to create a CAR that effectively regress the neuroblastoma in mice. Such peptides of monoclonal antibody generation and also epitope mapping of the CAR is generated in GenScript. In addition to generate peptides for clients, we understand the need of first identifying this neoantigen. And as such, GenScript has also formed a collaboration with Cayman to perform immunopeptidome profiling, which is a deep sequencing analysis of the MHC-associated peptides to identify neoantigens or cancer biomarkers and potential immunogenic sequence from MHC1 or 2. So human and tumor cell lies under optimal conditions for releasing intact MHC complexes. Using immunoaffinity chromatography, MHC-specific antibodies enrich the pool of MHC class 1 or class 2 from lysates. MHC-associated peptides are released under gentle eluting conditions and separated from captured MHC molecules. These peptides are then sequenced by LC-MS, MS, and then matched to the protein sequence databases or RNA sequencing data, as well as analyzed for mutations and post-translational modifications to facilitate the identification of potential neoantigens. So with also more than 16 years of peptide synthesis, we understand that sometimes neoantigens peptides are difficult to synthesize. For example, neoantigens can range 8 to 50 amino acids long, and peptides of more than 20 amino acids are challenging to synthesize. So at times, the neoantigen peptide selected may have a neutral charge, making them difficult for purification and their synthesis challenging. So hydrophobic peptides are also notorious for synthesis due to its insolubility in water, making purification difficult. In addition, these hydrophobic peptides tend to aggregate too. In view of this, GenScript has also been constantly developing new technologies and algorithms to aid in the synthesis of difficult peptides. We have the new algorithm for us to determine how difficult a peptide to be synthesized based on the length and aggregation and hydrophobicity and also isoelectric point. And the scientists in GenScript will determine the type of synthesis most suitable for the peptides. So this is an example of the new algorithm predicting the PI, the hydrophobicity, and also the difficulty in synthesizing a particular peptide and the recommended peptide synthesis method. We also have the Hyacine process specifically developed to synthesize difficult peptides. GenScript rigorously optimized the reaction temperature condensation reagents to increase the coupling efficiency. This allows us to have a higher success rate of 91% even for the hydrophobic peptides. We're also the first CRO to have double coupling technology, in which we use the coupling reagents from Hyacine and applied with microwave technology, which we have optimized the reaction conditions for. We were able to increase our success rate synthesizing previously unable to synthesize peptides at high purity. We also understand that many of these neoantigens will be required for validation in T-cell assay. So not only we have the neopre and Hyacine to ensure success of peptide synthesis, we also care about the quality of our peptides. So such as at the endotoxin level and also the TFA, which are crucial in T-cell assay. We have endotoxin control guarantee at 0.01 EU per microgram, purity ranging from crude to 98%, and also TFA removal to as low as 1%. Our fast synthesis as fast as 5 days are also one of the highlights of our services. In GenScript, we provide not only services, we also have analysis tools for peptides readily available free online. We have peptide analyzing tool, molecular weight calculator, and also a solubility guide. And we also have peptide library design for maybe T-cell epitope mapping. So as shown earlier, peptide libraries are good with identifying target-specific epitopes for T-cell, or even finding the perfect peptide conjugate for your gene therapy. So thus, choosing the peptide library appropriate for the scientific question to be answered or the applications is crucial. So in GenScript, we have 7 online library design tools for you to use. So for example, a truncated peptide library can be used to identify T-cell epitope. Optimal conditions for this kind of libraries are short peptide sequences from 8 to 11 amino acid on the MHC1 complex and 15 to 24 amino acid on the MHC2 complex. So application include testing of all possible T-cell epitopes across a protein of interest. So to better illustrate on the T-cell truncated peptide library, Yoshihiko et al. Published in the Journal of Immunology had demonstrated how T-cell truncated library can deduce the binding epitope of T-cell. First, using a T-cell truncated peptide library to identify the epitope region, they subsequently used different peptide libraries, such as random library here and also positional scanning library on a number of residues to identify crucial residues to enhance T-cell activation. So with all the information put together, I hope I have shared some ways in how one could enhance the cancer treatment using peptides and neoantigens in gene and cell therapy. Thank you.
Thank you, Dr. Leung, for the great presentation. So for those who have any questions, you can start to input your questions at the question box over here. We are running a bit short of time, but we still have like maybe 5 minutes to answer the questions from the floor. Okay. So the first question from the floor, what are the tips for designing an effective Ssdna template? So maybe Dr. Edward, you want to take this question?
Yep, sure. So in order to design an effective Ssdna template, for point mutations, it is suggested to use an asymmetric Ssdna design. And for large gene insertions, the homology arms with the 300 and 1000 base pair flanking the insertion genes have also been reported. So in most cases, the 500 base pair long of the homology arm should actually work. And also, it is important to design the Ssdna template containing silent mutations to mutate the sgRNA PAM sequence in order to avoid any of the secondary cleavage. Yep.
All right, thank you, Dr. Edward. So the next question, what's the recommended homology arm length on each side of the template DNA when designing an Ssdna?
Yep, as I mentioned earlier, so in most of the cases, the homology arm should be about 500 base pair in length.
Okay, thanks a lot, Dr. Edward. The next question, is there any tips on improving CRISPR knock-in efficiency?
Yep, the CRISPR knock-in efficiency is always the main concern to majority of the researchers. So in order to have this improve the CRISPR knock-in efficiency, the researcher should carefully design the sgRNA. So, and the sgRNA cleavage site should be close to the mutation sites, and it is best to be within about 15 base pair of each other. And also to optimize the transfection efficiency before transfecting the CRISPR raw materials and also, as well as the donor template. And also the researcher need to utilize some instruments like FACS sorter to enrich the transfected cells, and also even to use the selections marker to select the knock-in clones. That's about it.
All right, thank you, Dr. All right, thank you, Dr. Edward. So the next question, maybe Dr. Leung will need to take this. May I know if the neoantigen detected is only unique to one patient, or we need to find overlap mutated genes across few patients for the neoantigen candidate?
Usually, because the new antigen is from several patient samples, so actually we are actually gearing towards very personalized kind of medicine, but of course it will be again validated using T-cell assay that such peptides are actually common for the different patients. Yeah, but individually, yes, it will be for individual if it is an individual sample.
Okay, thanks, Dr. Leung. The next question is also related to the neoantigen peptide. Could you explain how much low level can we detect the peptides by LCMS technique? LCM-SMS technique, I'm so sorry.
How much low level? It depends on the HLA that is being expressed on the tumor cells itself. So some are they are more abundant. Then of course the sensitivity for the it will be increased.
Okay, thanks, Dr. Leung. Another question which is also related to neoantigen. What's the difficulty in synthesizing neoantigen?
Neoantigen, some of the difficulties involve sometimes it can be of varying length from 8 sometimes to even longer, example 50 amino acids. So for peptide synthesis, longer than as long as 50, it is quite the difficulty will increase because it is the additional of amino acid. There must be care that has to be taken, that this peptide do not aggregate or do not fold back such that it will hinder the additional of the subsequent amino acid because it is a chemical synthesis. So not only so, there will be also sometimes because it is a neoantigen. Sometimes it can be hydrophobic sequences. So hydrophobic neoantigens are also difficult for synthesis because sometimes they are actually insoluble in water, making purification very difficult. So these are some of the issues with difficult to synthesize neoantigens.
Okay, thank you, Dr. Leung. We are running out of time, but let's take on the last question from the floor. So do neoantigens show any cross-sensitivity?
Neoantigens are actually supposedly very specific because they are actually if I didn't understand your question wrongly, they're actually very specific to the tumor cell type. Unlike the tumor associated antigen, whereby there are still chances that it can be expressed in normal tissue. Neoantigen is a mutation, a somatic mutation for tumor cells only.
Okay, thanks, Dr. Leung. So due to time constraint, we'll just end the Q&A session here. For those questions that we didn't get to answer just now, I will email the answers from our speakers back to you. So thank you once again, Dr. Edward and Dr. Leung, for the great presentation, and to all of you who joined us today. And I hope you find this session today beneficial to your work. Thank you once again, and have a nice day. Bye-bye.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye.